What's up, YouTube? It's Subok Frakatomine. Oh, it's not even starting yet. Whatever. I need to make a small change on the YouTube. YouTube, a bit too small change. How do I get the brackets? Oh shit. Okay, that's not equal to. What was that? 4, 5? No. 5, 6? Yeah. Okay, it's streaming on YouTube, right? It is streaming on YouTube. YouTube stream is going on. I believe, yeah. Yeah. Yeah, 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 yeah. I hate this edit video of the first. Shut the fuck up, bro. Shut the fuck up, bro. Got it. Got it. Got it. Got it. Got it under control. So, what are the next topics? Most types of most important types of RNA. It was mRNA, messenger RNA, tRNA, transform RNA, and what was the tRNA? rRNA, ribosomal RNA. Make up the structures of ribosome along with proteins. Catalyzes peptide bond formation. Actual site of it's the actual site of protein synthesis. Optional bonus RNA, snRNA, involved in mRNA splicing inside the nucleus. miRNA/siRNA regulate gene expression by silencing mRNA. Who makes these mRNA? All the three RNAs are made from the DNA by the DNA. Made from DNA by the process called transcription. Transcribed by enzyme RNA polymerase. Depending on the gene, the product will be mRNA for making proteins, tRNA for translating mRNA during protein synthesis, rRNA for building ribosomes. From DNA templates. Okay. I get it. Now sketch how translation process at ribosome and label main important components. Let's see how these two AIs sketch. Protein synthesis. Transcription. It's the video I need. I mean, this person is getting revenue, right? Yeah. I wish this guy gets revenue because this guy helped a lot. Not like this guy.
Ja, einen schönen guten Morgen. Ich sehe schon, dass die
Why
Weltstädte
is my
Köln natürlich auch.
In this video, we're going to talk about transcription and translation. And here is just a basic overview of these two processes. Transcription is the process by which DNA gets converted into mRNA, also known as messenger RNA. And translation is the process of converting the information stored in messenger RNA and using it to build protein. Now, if you recall, which organ now is used to make proteins? What would you say?
Organ?
The answer is the ribosome.
Organelle,
The ribosome
okay.
is the site at which proteins are manufactured in the cell. So we're going to talk about that shortly. But let's go over transcription first before we talk about translation. Transcription occurs in 3 steps: initiation, elongation, and termination. Now, during initiation, RNA polymerase binds to the promoter region of DNA. Now, what is the promoter region? The promoter region is basically a short sequence of DNA. In eukaryotic DNA, it's TA also known as the Theta box.
Theta, okay.
Now, this sequence is located 25 nucleotides upstream of the site where transcription begins. The next thing that RNA polymerase does is it causes the
if the Theta box was is in the Indian accent, it would be Tata box.
Upstream of the site where transcription
Interesting.
begins.
I didn't get it. What?
That RNA known as the Theta box. The promoter region is basically a short sequence of DNA. In eukaryotic DNA,
Oh, that's how it is.
also known as the Theta box.
Tata box.
Now, this sequence is located 25 nucleotides upstream of the site where transcription begins. The next thing that RNA polymerase does is it causes the 2 DNA strands to separate. And during elongation, it begins to add nucleotides to the growing mRNA strand that we see here. Thus, RNA polymerase synthesizes mRNA starting from the 5' end going to the 3' end. However, RNA polymerase, it reads the DNA strand in the 3 to the 5' direction. Now, there's 2 strands that you need to be familiar with. The first one, the one that's used to synthesize mRNA, is called the template strand, or the antisense strand. So that's the one in which RNA polymerase is active upon. The other one, which is not used, this is called strand. So that's the one in which RNA polymerase is active upon. The other one, which is not used, also known as the sense strand. Some textbooks will call it the coding strand. Matches up with RNA, except the fact that uracil is found in RNA, but thymine is found in DNA. The last step of transcription, which is the termination step, the RNA polymerase molecule, the mRNA strand, they all separate from the DNA template strand. Poly A polymerase enzyme, it caps the 3 end of the mRNA strand, and this is known as the poly A tail. Now also, during the beginning of transcription, the 5 end is also capped. And the reason for this is to protect the mRNA strand from being degraded by certain enzymes. At the end of the termination step during transcription, DNA has been used to create a pre-messenger RNA strand. Now, this particular strand has something known as introns and exons. Introns are basically longer sequences of nucleotides that do not code for anything. So these, they must be removed in a process known as RNA splicing. Now the exons, those are shorter sequences of nucleotides, and they're going to be synthesize proteins. And so the exons removed. So now let's work on a practice problem. A sequence of nucleotides on a DNA strand. You're asked to write the corresponding sequence on an mRNA strand. What would it be?
Oh, RNA.
Feel free to pause the video and try it.
C,
So if we're reading the DNA strand
G,
from the
U,
3 to 5
A,
direction,
U,
we're going to have
C,
to write the corresponding
U,
mRNA
A,
sequence
U,
in the 5
A,
to 3 direction.
U,
So what
G,
letter
C.
corresponds to G? It's important to know that G always corresponds to C, and vice versa.
C,
C
G.
corresponds to G.
U.
Now, what letter corresponds to A?
U.
A usually corresponds to T, but there's no T in RNA.
Yeah.
Instead, A is going to correspond to U for uracil. But T in DNA corresponds to A in RNA. And so if we continue, everything else is going to be U, C, A, U, A, U, G, C. And so this is the mRNA strand nucleotide sequence in DNA listed above. Now let's talk about translation, which is the process of taking the information stored on an mRNA strand and using it to construct a protein. So once the mRNA strand is synthesized in the nucleus, it leaves the nucleus and enters the cytosol, where it interacts with a free ribosome or one that is attached to the rough ER. Now within the ribosome, it's going to interact with a tRNA molecule or a transfer RNA molecule. Now let's focus on the sequence of nucleotides on the mRNA strand. Notice that they're separated in sets of 3. Each set of 3 nucleotides represents a codon, which matches up with another 3 nucleotides on a tRNA molecule known as an anticodon. And each codon matches up with a specific amino acid. And so that's how the information stored in the mRNA strand can be used to construct a specific protein. And we're going to talk more about this later in this video. Translation, like transcription, occurs in 3 steps: initiation, elongation, and termination. So here in this picture, we have a ribosome, which is composed of 2 subunits, the small subunit and the large subunit. And the ribosome has 3 active sites: the E site, the P site, and the A site. Now, during initiation, we have the start codon AUG, which corresponds to the anticodon UAC. Now, that particular tRNA molecule has the methionine amino acid attached to it. And this tRNA molecule, it enters the ribosome at the P site, also known as the peptidyl site, where the peptide bonds are formed. Now, during the second step of translation, that is,
I don't get it. What? What the fuck?
Small sub. And so that construct step. Translation, like transcription, occurs in 3 steps: initiation, elongation, and termination. So here in this picture, we have a ribosome, which is composed of 2 subunits, the small subunit and the large subunit. And the ribosome has 3 active sites: the E site, the P site, and the A site. Now, during initiation, we have the start codon AUG, which corresponds to the anticodon UAC. Now, that particular tRNA molecule has the methionine amino acid attached to it. And this tRNA molecule, it enters the ribosome at the P site, also known as the peptidyl site, where the peptide bonds are formed. Now, during the second step of translation, that is during elongation, another tRNA molecule enters the A site. So I'm just going to draw it here. And during that process, a covalent bond will form between the two amino acids that we see here, highlighted by the red circles. Now, as the process continues, the tRNA molecule in the P site will move to the E site. And as it does so, it's going to lose an amino acid. And so what's going to happen is we're going to have a growing chain of amino acids that will leave or extend out of the ribosome. So make sure you understand that the tRNA molecules, they enter the A site and then they exit from the E site. And in the process, the amino acids are being joined together. So over time, this polypeptide chain is going to grow.
To form a protein. Ah,
Creating a
shit.
protein. The last step of translation, the termination step, begins when a stop codon is read. Now, there are 3 stop codons that you need to be familiar with. The first one is UAA, and then the second one is UAG, and the third one is UGA. Now, these stop codons, also known as nonsense codons, they don't code for any specific tRNA molecule. Instead, when these are read at the A site, they cause a release factor to enter the A site, which causes the small and the large ribosomal subunits to basically disassemble. So this is where translation ends, and the protein, it leaves the ribosome, where it eventually goes to the Golgi body for further processing and modification. At the Golgi body, the proteins undergo folding, where they form a specific shape to perform a specific function. And that's basically it for this video. Hopefully it gave you a good overview of transcription and translation. So that's all I got, specific function. And that's basically it for this video. Hopefully it gave you a good overview of transcription and translation. So that's all I got. Thank you.
Amino acid, amino acid, polypeptide. Nah, bro. I don't think I don't think ChatGPT got it. Amino acid, P site peptidol hold incoming tRNA with new amino acid, holds tRNA carrying the growing peptide chain, E site exit the tRNA. Okay, let me just check the last question. Have to sharpen everything I know. Chaperone is a protein that assists in proper folding, assembly, stabilization, or degradation of other proteins. Okay. Prevents misfolding, shield newly synthesized proteins from aggregation, refold damaged proteins, helps protein regain their functional 3D structure after stress. Assist in degradation. Tag misfolded proteins for destruction by proteasomes. Examples of well-known chaperone: heat shock protein 70, HSP70, binds to exposed hydrophobic regions of unfolded proteins, preventing aggregation, uses ATP to fold client proteins or deliver them to other chaperones. Induced by stress elevated during heat shock, hence the name. Deliver them to other chaperones. Induced by stress elevated during heat shock, hence the name. I think that's the best video.
You want to have the camera with the best color? Lumix is the one. I'm using Lumix cameras for...
In order to become a useful protein, the polypeptide produced by a ribosome during translation must be folded into a unique three-dimensional configuration and must bind any cofactors required for its activity. It might also need to be modified appropriately by protein-modifying enzymes, such as are not only a waste of energy for the cell, but they are also dangerous. These aberrant proteins typically have exposed hydrophobic regions, which cause them to clump together into aggregates, which can accumulate and cause severe diseases. The cell has two lines of defense against the formation of these dangerous misfolded proteins: molecular chaperones and the proteasome. Molecular chaperones help proteins fold correctly, while the proteasome destroys misfolded proteins through proteolysis. The molecular chaperones and proteasomes compete over these proteins. The longer a protein remains misfolded, either because chaperones haven't reached it or because they haven't succeeded in fixing it, the more likely it is that a proteasome will degrade it. Let's discuss chaperones first. Chaperones work by binding to exposed hydrophobic patches on misfolded or incompletely folded proteins and hydrolyzing ATP. Some of the energy expended by chaperones is used to perform mechanical work, but much more is used to ensure the accuracy of protein folding. The proteins that chaperones help fold properly are called client proteins. There are several families of molecular chaperones in eukaryotes, which function in different organelles. The ones we will be discussing are HSP60 and HSP70, each of which has...
Which was DeepSeek talking about, 70.
...of associated proteins. HSP stands for heat shock proteins because lots of them are produced when a cell is exposed to elevated temperatures, responding to increased amounts of misfolded proteins. HSP70 acts early on in the process of protein folding, binding to polypeptide chains emerging from ribosomes where there is a chain of 7 hydrophobic amino acids. HSP70 is aided by HSP40s, and many cycles of ATP hydrolysis are generally required to fold a single polypeptide chain correctly. HSP60 acts on fully synthesized proteins, forming a large barrel that serves to isolate the protein and provide better conditions for folding. HSP60, which is a type of molecular chaperone called a chaperonin, initially captures proteins along the rim of one of its barrels. Then, the binding of ATP and a protein cap increases the diameter of the barrel and partly unfolds the protein. Inside the barrel, the protein has a chance to fold in a more favorable environment. 15 seconds after ATP binds to the HSP60, it hydrolyzes, weakening the complex. The binding of another ATP molecule makes the protein leave. If the protein is still not folded correctly, it can repeat the cycle. Proteasomes are so important that they constitute almost 1% of cell proteins. They are dispersed throughout the nucleus and cytosol. If a protein folds correctly and quickly, then none, or only a small bit of it, gets degraded. A slowly folding protein is at risk for a longer time, and more of it might get destroyed before the remaining amino acids attain the correctly folded state. I will discuss the destruction of proteins in detail in the next video. If you enjoyed this video, like and subscribe. You can also support me by following the link to my...
On Fiverr, you can find the perfect freelancer for your digital marketing. But you probably already know a guy who knows a guy who has a cousin who has a neighbor who can do it. Well, we also know a guy, David, a freelance digital marketer with tons of reviews like this. We also know Kate.
Proteins are made up of folded polypeptide chains, which are composed of 20 different amino acids, each with different chemical properties that are linked to each other via covalent peptide bonds. The sequence of atoms repeating to form the peptide bonds is called the polypeptide backbone. The side chain of each amino acid, which is the part that makes it different from the other 19 amino acids, can be either polar or nonpolar. The polypeptide chain can fold in many different ways called conformations. Conformations are the spatial arrangement of atoms that can result from movement of atoms without breaking of bonds. Protein structure can be described in terms of 4 levels of organization. Primary conformations. Conformations are the spatial arrangement of atoms that can result from movement of atoms without breaking of bonds. Protein structure can be described in terms of 4 levels of organization: primary, tertiary, and quaternary structure. Is simply the amino acid sequence of the. Important to remember that this sequence contains all the information necessary. The secondary structure is formed by hydrogen bond interactions of adjacent amino acids. Large numbers of such local interactions form alpha helices and beta pleated sheets. Shape. Large proteins often consist of several protein domains, which are distinct structural units that fold somewhat independently from one another. Quaternary structure is found in those proteins that have 2 or more interacting polypeptide chains, which are then termed subunits. Secondary structures include alpha helices and beta sheets. An alpha helix occurs where a polypeptide chain coils like a spring, with 1 turn every 3.6 amino acids. In alpha helices, the NH of a peptide bond is hydrogen bonded to the C double bond O of another peptide bond, which is 1 coil up in the helix structure. Note also that all the NH groups point in one direction, towards the N terminus, and all the C double bond O groups point in the opposite direction, towards the C terminus, and this is what gives the polypeptide chain polarity. The C terminus is partially positively charged, while the N terminus is partially negatively charged. Beta sheets can form parallel chains, which are made from neighboring chains running in the same direction, or anti-parallel chains, which are made from itself so that nearby sections. While hydrogen bonds in an alpha helix are bonds in beta. Tertiary structure formation by the pattern of which plays a central role in determining the protein's final molecules, such as the nonpolar side chains, are entropically driven together in an aqueous environment, limits their disruption of the hydrogen bonding of surrounding water molecules. As a result, nonpolar amino acids tend to be found predominantly in the amino acids face the outside of the protein, forming hydrogen bonds with one another and with the water molecules around the protein. Those polar amino acids that are on the inside of the protein bond with one another or with the polypeptide backbone. It is thought that protein folding happens roughly along the following lines. Secondary structures form first. Hydrophobic collapse, during which nonpolar amino acids aggregate, happens next. Long-range interactions between secondary structures cause further folding to occur. Throughout this process, there may be 1 or more intermediate states, such as what has been termed a molten globule. As a final note, there are 3 basic classes of proteins, which are distinguished based on shape and solubility: globular, fibrous, and membrane proteins. Globular proteins are spherical in shape, with as little surface area per volume as possible. These proteins are marginally stable, and this marginal stability facilitates motion, which in turn enables function. Hydrophilic amino acids occupy this small surface area, making these proteins highly soluble in water. Fibrous amino acids are simple linear structures which have structural roles and are insoluble. Lastly, membrane proteins are associated with cell membranes. If you like this video, like and subscribe.
Okay.
You can also support me by following the link to my Patreon.
I mean,
If you
this
have any
is
topics you'd
basically
like me
how
to
the cell makes itself. What is a protein? What? Oh, yeah, it's me.
called molecular chaperones. That are some specialized
Chaperones?
proteins that
Chaperones.
help in folding and do many other functions, which we are going to see in this video. These chaperones are a part of quality control system, particularly the protein control system, which is the mechanism by which a cell monitors proteins to ensure that proteins are in a stable and in a native state. The first important function of chaperone proteins is that they assist in protein folding and also rescue the misfolded protein back to its normal conformation. Second thing is that chaperones prevent the protein degradation. It's obvious that when the protein is correctly folded, it will not be sent to proteasome degradation, thus prevents the protein degradation. Then we have another class of chaperones that prevent the protein aggregation. Or their activity is in a way that they take out the proteins, they rescue the proteins from aggregated states. That is done by a class of chaperones called disaggregases. When all the explained functions of chaperones are well up to the mark, there will be no prion formation, proteasome degradation, the process and gets aggregated, and the final way for protein is to go into the proteasome system, which we are going to see later on what this proteasome system is. First of all, we have a linear amino acid sequence which needs folding to get it functional. And this process of protein folding proceeds via an intermediate state called the molten globule state. Under normal conditions, the molten which needs folding to get it functional. And this process of protein folding proceeds via an intermediate state called the molten globule state. Under normal conditions, the molten globule state is transformed into final shape, that is the native state of protein. But sometimes the intermediate state skips the pathway and goes off the fold, and this ubiquitin mediated pathway leads to
Destroy.
protein degradation. And in this way, the misfolded protein, the damaged protein, is eliminated. And there are some aggregated proteins that resist the proteasome system for degradation. And that proteins later on become the prions that leads to the several disease later on. Now let's see the types of chaperones. The majority of chaperones are from a family of HSP, that's heat shock proteins. These heat shock proteins are produced or translated during the proteins later on become the prions that leads to the several disease later on. Same functions but with different names. Then we have another class of chaperones called lactin chaperones, which includes two chaperones. One is calnexin, other one is calreticulin. Both are mediated via calcium IP3 pathway. Both chaperones have function in immune system also, like antigen presentation processing in MHC class 1 molecules. Then we have GRP58 protein, that is glucose regulated protein chaperone that assists in glycoprotein folding. Another protein folding chaperone is PDI, that's protein disaccharide isomerase. On the basis of chaperone activity, we categorize the chaperones into three classes. First one is the foldases, second one is the holdases, third one is the disaggregases. Foldases, as the name implies, are the chaperones that assist in folding, and also they transform the misfolded protein back to the native conformation. And these foldases require ATP, so they are ATP dependent. The example of foldases is HSP60 or GROW-EL in E. coli. Then another class is holdases. It prevents protein damage by providing favorable conditions. Unlike foldases, these holdases do not fold the protein or do not correct the misfolded protein, but they prevent the protein. And by the help of foldases, when the favorable conditions arrives again, the holdases supply back the protein to the foldases for folding process. Then finally we have disaggregases. These are the chaperones that prevents aggregation, prevents prion formation, or rescues proteins from aggregated states. And one of the example of disaggregated chaperones is HSP104 protein that is found in yeasts. So this is all about
Yeast,
chaperones, the
okay.
function of chaperones, the types of chaperones. In the next video, we'll be discussing about the mechanism of these foldaces, particularly the HSP70 and HSP60 machinery. Thanks for watching the video. You can
How old is it? 2017.
My work is easy because I use ClickUp. Here's how I keep track of all my tasks. I start by brain dumping everything I need to get done in a list view. Next, I set a priority.
Towels, shirts. Let's just say I invest a lot in anti-wrinkle laundry spray. Amazing invention. My issue with folding extends to paper too. I know foldables in the classroom can be a powerful way to organize concepts, but for me, it was the actual folding part that I tended to get stuck on. You may think of folding as a convenience, of a way to take something and make it more organized or condensed so it doesn't have to take up so much space. This is true, but in biology, folding can actually have a lot to do with function. We've mentioned how amazing proteins are. They can play so many roles. They can make up channels, be part of a structure, serve as enzymes for important biological processes, be involved with protecting the body, just to name a few. We've also mentioned that you are making proteins all the time in a process known as protein synthesis.
Oh yeah,
But the
shit, that's
conclusion
right.
of producing a long chain of amino acids
Bodies keep
doesn't
processing
necessarily equal
proteins.
a functional protein.
Oh
There are
shit.
modifications to a protein that often need to happen in order for it to be functional. By modifications, we can mean many things. It might be adding certain chemical groups, such as phosphorylation, something to definitely explore. But another important event to make a functional protein is, believe it or not, folding. But before we get into protein folding, let's talk about shape and why shape is so important. Shape and function in biology frequently go hand in hand. In our cell signaling video, we mentioned how protein receptors and the signal molecules that bind them can fit together so perfectly to start some type of cellular response. Or in our enzyme video, we talk about how enzymes, which are frequently proteins, have a very specific shape for the substrates that they build up or break down. When we talk about the way proteins are folded, we need to understand the different levels of protein structure because there are different ways of folding that can happen in the different structural levels. The first level of protein structure is primary structure. This is the sequence of amino acids that make up a protein. Amino acids are the monomer, which means the building block, of a protein. They are held together by peptide bonds. In protein synthesis, amino acids are added to form a polypeptide chain, and proteins are made of one or more of these polypeptide chains. Genes, which are made of DNA, determine the order and number of these amino acids. That sequence is critical. In our mutations video, we talk about how one amino acid in sickle cell disease, even as has the potential to affect a protein's point out, each amino acid has a carboxyl group, an amino group, and an R group. An R group is also called a side chain. So even though we have them drawn here like a chain of circles, realize that each of these circles we're drawing is an amino acid like this. Next, we move on to secondary structure. Folding is really going to start to happen. In secondary structure, the sequence of amino acids that we mentioned in primary structure can fold in different common ways. Which one of those foldings the protein does depends on the amino acid, largely in part to hydrogen bonds. Those can occur at specific areas of the protein. These are hydrogen bonds involving the backbone of the amino acid structure. Onto tertiary that occurs in the 3D shape of a functional protein due to something we haven't mentioned much. Side chains. See, the amino group and the carboxyl group are generally standard parts of an amino acid, although the R group found in amino acids can vary among different amino acids. That means the R group can define the amino acid and can make amino acids behave a certain way. For example, some R groups are hydrophilic. They like water. Some R groups are hydrophobic. They don't.
Hydrophobic.
And remember that proteins contain many amino
Hydrophobic.
acids, which can
Hydrophobic,
contain
same
different
thing.
R groups. and so different
No,
areas of the protein can therefore
no,
be
it's not the
impacted
same.
based on those R groups. When protein folding is going on, amino acids with hydrophilic R groups may hang out on the outside, while hydrophobic R groups, where are they? They might hang out at the inside part of the protein. The 3D shape is due to other interactions besides hydrophobic interactions. Ionic bonds, van der Waals interactions, disulfide bonds, and hydrogen bonds, all involving the R groups, can influence the folding occurring in tertiary structure. Something to explore. Now, when we've been talking about a protein, we've been talking about a polypeptide chain that has been folded into a functional protein. But proteins can be made of one or more polypeptide chains. And in quaternary structure, you are looking at a protein consisting of more than one polypeptide chain. Each of these polypeptide chains can be a subunit, and interactions between them, such as hydrogen bonds or disulfide bonds, can keep them together. Going back to folding, I know what you might be thinking. Who is doing this folding anyway? Are the proteins just folding themselves? Well, the interactions mentioned, like hydrogen bonds and R group interactions, are occurring depending on the protein's own amino acids. One reason why amino acid sequences are very important for protein function. But folding is far more complex than that, and there can be intermediate steps involved when a protein is folding. In fact, there's a phrase you can search called the protein folding problem to learn more about the questions scientists continue to explore regarding protein folding. Research has shown that proteins often have help in the folding process. Chaperonins, for example, are proteins that can help with the folding process. They have almost a barrel shape. Proteins go into them, and the chaperonin tends to have an environment that is ideal for the protein's folding, and this can help the protein be folded correctly so it's functional. Just wish I had something like that for my towels. All of these interactions we mentioned in primary, secondary, tertiary, and quaternary structure are paramount for a mature protein to have its correct shape so it can carry out its function. And that's very relevant. There are many diseases that are related to protein misfoldings. Check out some of our further reading suggestions in the description about that. One last thing we haven't mentioned. Each protein has an ideal environment for functioning, which might include a certain temperature or pH range. If the protein is exposed to something outside of its ideal temperature or pH range, exposed to high heat, for example, you can disrupt the interactions that we had talked about taking place at the different structural levels. This can denature the protein, which disrupts its shape. This prevents it from functioning correctly. And depending on what caused it to be denatured, sometimes you are interfering with many levels of protein structure. Sometimes it's just one or two levels. Sometimes denaturing a protein may be reversible. The environment that a protein is in definitely matters for its functioning. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Hell yeah. Chaperone. So these are proteins that help in folding, aggravating, or degrading a protein. It's really interesting. There's a lot of shit happening inside a really small thing. So these are the example questions. What do we have here? Levels of protein structure. These chaperone. So these chaperones are also made by the stuff we talked earlier, like transcription. Yeah, I mean, yeah, it's a. Mhm. Yeah, yeah, I get the stuff. This guy is the. Oh, you got a structure. It's a barrel. Let me wash my hands. We got a lot of stuff to. I mean, I still haven't figured. I mean, I just know in a really vague way of what these answers for these questions are. I need to go in deep because I know for a fact these are not these are just to know. You know, these questions are just to know what I should look into the subject. For the exam, it's not gonna be like this, and for myself, I need to learn more of this. This is what it's all about. Oh, come on. ഹലോടാ എടാ നീ റൂമിലാണോ ആ നിന്റെ ഒരു സിങ്കിന്റെ ഭാഗ്യമുണ്ടോ ചോദിക്കണമെന്ന് വിചാരിക്കില്ലേ ഹലോ വിചാരിക്കുന്നത് എൻ്റെ ഒരു സിക്കിന് വെക്കണ്ട. അല്ലല്ല, സിക്ക് സിക്ക്. ഞാൻ താഴോട്ട് വരാം. അതെ, എവിടെ? ആണോ, നീ ഇതിലോട്ട് വരണം. ആണോ, സെറ്റ്. ഓക്കെ, ഞാൻ Yo, my people, I'll be right back. I'm back. I'm back strong and powerful. So, how much time did I waste today? How do I know? Did I waste today? Damn, half of the time was not here. Half of the stream I was not here. I'm sorry, people. I just went outside to get the smoke. Transcribed into mRNA by RNA polymerase during heat shock. The transcription factor HSF1 heat shock factor 1 activates chaperone gene expression. Mm-hmm. So this, so for example, common okay, I gotta charge the pencil. Dude, I'm hungry. %30%. That's not a small number. A liver cell versus a neuron. So, in which mm-hmm. Oh yeah, this guy. It's the barrel. Nucleoplasmins. Sperm cells repurpose chaperone. Ah. No, now 1 and 2. First line defender against misfolding mechanisms. Binds to the exposed. Multi-subunit barrel. Small, medium-sized proteins, large, complex proteins. Folding sides, binds externally, encapsulates in central cavity. Binds, releases. So all these are happening in the cell's cytoplasm. So people, it's getting dark and I gotta eat. I will wake up tomorrow and I should be looking into it tomorrow. I should be completing a lot within tomorrow. I need to get back my grind. I'm still not taking this serious. So peace out people. Peace out YouTube. I respect everyone for supporting me. Appreciate it. Oh.